Hi, my name is Didi Jimenez, and today I'm going to go over the structure of skeletal muscle. So first, let's start with the basic introduction. As we know, all activities having to do with movement require muscles. We use our muscles for locomotion, our upright posture, balance, support of our internal organs, production of heat, and for controlling valves and body openings, just to name a few. Our body consists of three main muscle types: skeletal, cardiac, and smooth muscles. They are categorized first by their appearance, aka having alternating dark and light bands, or striations. Our striated muscles are cardiac and skeletal muscles, while our unstriated are our smooth muscles. Secondly, our muscles are categorized on whether or not they are innervated by the somatic nervous system or if they're innervated by the autonomic system and not subject to voluntary control. Our skeletal muscles are voluntary, while our cardiac and smooth muscles are involuntary. So moving on to our main topic, which is our skeletal muscle structure. Our skeletal muscles are attached to bones by tendons, and together they are all responsible for all body movement. Fun fact, about 40% of a man's body weight is skeletal muscle, and for a woman, it's about 32% of body weight. Okay, now moving on. One single skeletal muscle cell, aka muscle fiber, contains many muscle fibers lying parallel to one another and are bundled together by connective tissue. As we know, our skeletal muscle are striated, meaning they are made up of fibers with a cross pattern of red and white lines that usually extend the entire length of the muscle. These skeletal muscle fibers are formed by myoblasts that are fused together during embryonic development, resulting in visible nuclei dispersed within the plasma membrane. These muscles also have an abundance of mitochondria, aka energy-generating organelles, and a lot of myofibrils. Myofibrils are specialized contractile elements that consist of cytoskeletal microfilaments, which are either thick or thin. Thin filaments are made up of mostly actin, while thick filaments are assembled of the protein myosin. So overall, the level of organization is, first you have our whole muscle, which is an organ, our muscle fiber, the cell, a myofibril specialized intracellular structure, and then thick and thin filaments, or cytoskeletal elements, and then our myosin and actin, our protein molecules. Okay, now let's go a little bit more in depth. Within myofibrils, we can find alternating A bands, which are dark, and I bands, which are light. Keep in mind that all of these bands are lined up parallel to each other and are the result of alternate stacked sets of thin and thick filaments. They are geometrically precise due to cytoskeletal proteins. A bands are made up of stacked sets of thick filaments with portions of thin filaments that overlap on both ends of the thick filaments. The thick filaments within these A bands extend their entire length and only lie within A bands. Now, taking a look at our picture. Here we can see the different bands, lines, and zones. You can see I just mentioned the A band, which you can see extends this area. This is a very nice picture that shows all the areas. The H zone right here is a lighter area where thin filaments don't reach. Right here. aka only central points of thick filaments are found here. The M line we see here, I think it's like this little zone right here, the little pass you see, extend vertically down the middle of A bands within the H zone. These house our supporting proteins that hold the thick filaments together in a vertical direction within each stack. The I band, which is the remaining thin filament that doesn't project into the A band, here only consists of thin filaments from two adjacent sarcomeres, but not the entire length of these filaments. Next, we have our Z-line, which is located between each eye band and is made up of a flat cytoskeletal disc that connects the thin filaments of Z adjoining sarcomeres. Sarcomeres are the functional unit of skeletal muscle and are located between two Z-lines, as seen here. It's like the perfect picture for this. They consist of one whole A band and half of each I band, located on either side. Interestingly enough, for growth, a muscle increases by adding sarcomeres on the ends of myofibrils, but not by increasing their actual size. Titin, which is the largest protein in the body, is a single-stranded and highly elastic. This protein extends both directions from the M line along the length of the thick filament all the way to Z lines at the opposite end of the sarcomere. Opposite end. It serves three important roles. That's why I mentioned it. It serves as a scaffolding, as an elastic spring, and lastly, it participates in signal transduction. The last important thing for this slide is our cross bridges that extend from each thick filament toward the surrounding thin filaments in the areas where thick filaments overlap. Now, let's take a look at our thick and thin filaments. Thick filaments are formed from myosin molecules packed together in a certain arrangement. These myosin molecules are composed of proteins with two identical subunits, and they have like a golf club shape. These two units are intertwined with two globular heads projecting out at one end. There are two hinge points locations where these myosin can bend. That is along the tail and at the neck/junction of the tail with each head. The arrangement within thick filaments includes myosin molecules lying lengthwise with a staggered array toward the center or the tail, and protruding outward is the globular heads. These outward pointing heads form cross bridges between thick and thin filaments. At these cross bridges, you can find both an actin binding site and a myosin, ATPase, or ATP splits, site that are both crucial for the contractile process of muscles. Thin filaments, on the other hand, are composed mainly of spherical molecules named actin. It is important to note that not just actin is found within thin filaments. You also have tropomyosin and troponin. Now looking at them separately, let's start with actin molecules, which are formed by two intertwined strands that form the backbone structure for thin filaments. Actin binds to myosin at cross bridges, which results in contraction of muscle fibers. In a relaxed muscle, no contraction occurs due to the fact that actin can't bind with cross bridges because of the position of tropomyosin and troponin within the thin filament. Now, I believe in order to understand the mechanism, you have to know the parts. Tropomyosin are thread-like proteins that lie end to end alongside the groove of the twisted actin. These proteins cover actin sites that bind to cross bridges and blocks interaction and leads to contraction of muscles. Troponin, on the other hand, is made of three polypeptide units that bind to tropomyosin, actin, and Ca2+. If it doesn't bind to Ca2+, it is stabilized and tropomyosin in its blocking position over actin cross bridges binding sites. When troponin actually binds to Ca2+, it causes a shape change resulting in tropomyosin slipping from its binding position and therefore contraction of muscle due to the binding to actin. This means that tropomyosin and troponin are regulatory proteins because of their role in covering, aka preventing contraction, or exposing, aka allowing contraction, to the binding sites for cross-bridge interaction between actin and myosin. So why is this important, you may ask? Let's take a look at BMD, Becker muscular dystrophy, which is an allelic disorder of the dystrophin gene at the XP21 locus 1-2. Just to make things clear, dystrophin is found in both cardiac and skeletal muscle and plays a role in protecting the sarcolemma from mechanical stresses of muscle contraction. In this case, we have an 18-year-old male that was diagnosed with BMD and presented with chest discomfort, nausea, dilated cardiomyopathy, and dyspnea at rest. Keep in mind that BMD's primary cause is heart failure, and it can be characterized by reduced expression of the protein dystrophin. Okay, so moving on to our main case. This patient was examined in earlier years when he was 3 years old because of enlarged calves. For this, the patient underwent a muscle biopsy and a dystrophin gene analysis that owed to his enlarged calves and elevated levels of serum kinase without overt weakness. Many other tests were done, including a blood test that showed elevated serum levels of creatine kinase, an electromyogram that demonstrated myopathic changes that consisted of small polyphasic potentials, a biceps biopsy that showed dystrophic features, and lastly, a multiplex polymerase chain reaction that showed a deletion of exons 45 to 49. This analysis plays an important role for dystrophin encoding genes. Unfortunately, at the age of 18, the patient presented with chest discomfort, nausea, dyspnea at rest. Keep in mind, there was no significant family history of muscle disease. This occurred once the patient ascended the stairs properly and remained ambulant with mild proximal weakness. At the time the patient was admitted into the hospital, heart rate and blood pressure were normal, and no murmur or rails were detected. So what was going on with this boy? A series of tests had to be done to find out. He underwent a manual muscle test that showed minimal weakness of lower proximal muscles. The patient didn't struggle with standing from squatting position. However, a chest röntgenogram showed cardiomegaly, and electrogram exhibited an abnormal ST to T wave by atrial enlargement. This can be seen at row 2. It's sort of like a hump within a hump. I saw it like it was a bump, and then it kept going. It was a double bump. You can kind of see it. It's too blurry to see it, but it was like one of these humps that had the double hump within it. And then there was also a left ventricular hypertrophy visible at V6. V6 right here, it was like a backwards. So finally, after so much testing, doctors were finally getting somewhere. Unfortunately, there was no indications of inflammation and viral titters, so echocardiograms, both 2D and M modes, were conducted, and they showed severely dilated left ventricle activities with diffuse hypokinesis, as you can see on the right. Unfortunately, even with all these tests, the function of the patient's left ventricle deteriorated gradually despite intensive treatment for heart failure, leading to the patient's death due to congestive heart failure 5 months after initial cardiac symptoms. So how does this all play out? Let's discuss it. So the 18-year-old boy passed from early onset DCMP with BMD, Becker muscular dystrophy, which has many cases that suggest that cardiac dysfunction isn't clearly correlated with skeletal muscle severities. In BMD patients suggest that pathogenesis and genotypic correlations are possible. In this case, we can say that early onset DCMP and advanced heart failure were related to the deletion site of exons 45 to 49 in the dystrophic gene, meaning it is more sensitive in cardiac muscle dystrophin compared to that of skeletal muscle. Nonetheless, more studies have to be conducted to clarify the genotypic-phenotypic correlation of cardiac function in dystrophinopathy. These are our resources here in case y'all want to look further into it since we ran out of time. But thank you guys and see you on the next one.